For over a century, Alzheimer's disease has been considered irreversible. Consequently, research has focused on disease prevention or slowing rather than recovery. Despite billions of dollars spent on decades of research, there's never been a clinical trial of a drug for Alzheimer's with an outcome goal of reversing disease and restoring function. And now, a research team led by Kalyani Chaube, PhD, from the Piper Laboratory at the University Hospitals at Case Western Reserve University in the Louis Stokes, uh, Cleveland, Virginia, Cleveland VA Veterans Affairs Medical Center, has challenged this long held dogma in the field. They now demonstrate that Alzheimer's can be reversed by restoring the NAD plus balance or nicotinamide adenine dinucleotide levels in the brain. The team showed that the brain's failure to maintain normal levels of this central cellular energy molecule called NAD plus is a major driver of Alzheimer's, and that maintaining proper NAD plus balance can prevent and even reverse the disease. That is absolutely fantastic news for anyone afflicted with Alzheimer's. NAD plus levels decline naturally across the body, including the brain, with age. Without proper NAD plus balance, cells eventually become unable to execute critical processes required for proper functioning and survival. In this study, now, the team showed the decline in NAD plus is even more severe in the brains of people with AD and have used mouse models to find a breakthrough. While AD is uniquely human condition, it can be studied in the laboratory mice that have been genetically engineered to express genetic mutations that cause Alzheimer's in humans. The researchers used two of these models. One line of mice carried multiple human mutations in amyloid processing, and the other mouse line carried a human mutation in the tau protein. Amyloid and tau pathologies are two of the major early events in Alzheimer's, and both lines of mice develop brain pathology resembling Alzheimer's, including blood-brain barrier deterioration, axonal degeneration, neuroinflammation, impaired hippocampal neurogenesis, reduced synaptic transmission, and widespread accumulation of oxidative damage. These mice also develop severe cognitive impairment that resemble what is seen in people with Alzheimer's disease. After finding that NAD plus levels in the brains declined precipitously in both human and mouse Alzheimer's disease, the research teams tested whether preventing the loss of brain NAD plus balance before disease onset or restoring brain NAD plus balance after significant disease progression could prevent or reverse AD, that is Alzheimer's. In their previous work, published in the Proceedings of the National Academy of Sciences, USA, they showed that restoring the brain's NAD balance resulted in pathologically and functional recovery after severe, long-lasting brain injury. They restored NAD balance by administering a now well-characterized uh, medical agent known as P7C3-820 developed in the Piper lab. Not only did preserving NAD balance protect mice from developing AD, but delayed treatment in mice with advanced disease also enabled the brain to fix the major pathological events caused by the genetic mutation. Both lines of mice fully recovered cognitive function accompanied by normalized blood levels of phosphorylated tau 217, a recently approved clinical biomarker of Alzheimer's in humans, uh, suggesting that it's a positive confirmation of disease reversal and highlighting a potential biomarker for future clinical trials. Indeed, someday restoring the brain's NAD balance might help patients recover from Alzheimer's disease. That's what the experts say. The damaged brain can, under some condition, repair itself and regain function. The agent P7C3820 that the scientists use enables cells to maintain appropriate balance of NAD plus under conditions of otherwise overwhelming stress without abnormally raising NAD levels. The next step would be to identify which aspects of brain energy balance are most important for recovery, identifying and evaluating other approaches to Alzheimer's reversal. If you enjoyed this presentation, please subscribe. Thank you.